After.
Yeah.
I'm just gonna have that run. It'll likely work, and then when we do yours, which will be longer, it'll just be black screen when it's done recording. I tried everything to try to receive the codecs and then change my settings on the Android. Nothing works. I lost the video. Even the file size is more of an MP3 file size than an MP4, even though it says .mp4.
Can you split it into separate videos?
I'll split your seminar and then I'll split it and make a new one for the Q&A.
So, okay.
That's a good idea.
No, like,
Oh,
give.
there.
Mm-hmm. So I click it and it's like other users sign in.
I'm just gonna use the restroom real
Can you
quick.
not sign in with another account? Let me just find Joanna.
Yeah.
Hey, you should be able to sign in with any account.
Oh, like any temple account?
Yeah.
Oh.
It's just a tough computer.
Oh.
Seibold, Dr. Seibold works with you on NeuroVISOR, right?
Mm-hmm.
I tried applying for the lab a couple summers ago, but I didn't have a background in C or anything
Nope.
but VR background at the time.
Oh, okay.
So
I'm pretty sure.
Wait, no, you're saying
you write
nothing
to me
yourself,
or
you're
to-
saying
No,
it's-
to Dr.
it's-
Seibold.
it's-
I didn't know who was in charge at the time.
What am I doing?
Pictures like
No, like it says sign
Yeah,
in
a couple
for
of years
users,
ago,
so
that
it's
sounds like
definitely me.
that
So
was
I can sign in.
early on
Yeah, could
in
you?
the
Because like,
developmental phase, because the project's been running for maybe
Welcome.
5 years now.
Have you patented it?
No,
yes,
it's
please.
an open source project, so it's on GitHub. People can, if they have a VR headset, can.
Thank you. I don't know
See,
why.
yeah, we're
I
gonna
was
have a
thinking-
lot of questions, like, out of intrigue, just in general, as well
So what,
as
it's just
getting
like
into the
your-
guts
your
of it
normal
during your
TUID
presentation.
password?
Yeah, just you guys.
Yeah, I'll talk
Alright,
talk
thanks.
about
Hopefully it works.
the biology quite a bit,
yeah.
just to get the background of what it actually does, and then show a few examples.
It should be like 4, yeah.
Yeah, it's hard to like demo it without the VR headset, but yeah, so you'll hopefully get an impression of what it does.
And you still just, not just, I know it's a really big data set, but you still use solely neuromorpho?
Yes, yeah. I mean, any cell with a specific cell file format can be used.
Like top files, or more like,
Well, one of the standards is this SWC format, which is, the simple ASCII coded text file, from which we read the geometries, and I'll talk about it. The hard part is that everything that's on neuromorpho, for instance, is just a one-dimensional graph of the cell, and for VR we need surface meshes to visualize in 3D and interact with. So we've been developing a tool that generates those 3D geometries, which is a fairly non-trivial task.
is it PDE based?
The meshing is basically discrete geometry based.
Cool.
Yeah, but the actual simulations, they're PDE based.
I've noticed a lot of the work I'm doing now, either for the job or hobby, is really the math and CS techniques cross-fertilize each other. Or rather, they're kind of like the same, almost the same exact thing.
Yeah, computer science was math until computers were designed. Before that, it was all math, and then since computer science, that area of math became something, became its own thing. But historically, it was all math.
See, once I'm trying to trace the softwares, trace the structures with Autodesk, but then I found a way to transfer the geometry file when it's all nice and pretty into Unity.
Mm-hmm.
So they're doing a lot of that now on my side too, like using Unity Engine to navigate through structures and their dynamics.
They use Autodesk to generate the 3D models.
I start in lamps, and then I move on to
Well, I mean, we have
Blender
it's
with
just
their
installed,
geometry
apparently.
nodes package and their molecular nodes package for smaller objects. Then finally I put it into, I haven't done this part yet, but I put the geometry output out of Blender into Autodesk, and then I haven't done Autodesk to Unity yet, but I've seen preprints that show it's easier.
Okay.
Yeah.
And you start with labs, meaning you do molecular
But it's fine.
dynamics.
If this works, it can be Anthony for today.
And those you want to.
Oh, okay. Yeah, otherwise it's this, but we don't want to.
I mean, that would be beautiful.
Well, I will turn that to
Maybe I can ask you about that after you present. The other thing is, you know, for larger structures, I'm trying to use CFD, like OpenFOAM. I'd be curious if you could run an
Yeah.
OpenFOAM.
Well, I'm fine.
I'm just spitballing here.
Okay,
I'm curious if
I'll be here till
NeuroVISor
5:30.
can handle and navigate through like a hybrid OpenFOAM NeuroVISor simulation. I know it's not necessary for the size of the structure you're currently working with, but well, it's arguable, but.
Yeah, that, yeah, that's a whole different question, because we want to load an external tool during runtime. We're experimenting with that actually at the moment, with another tool, but yeah,
Only one person now.
that's still
Check again.
hypothetical to see how it could efficiently run within an active VR simulation. Can you load and run external tools efficiently?
Hi, everybody. Welcome. Thank you for coming. Today I'm honored to welcome Dr. Gillian Queisser. Got it. To speak with you all here at Studio today. Dr. Queisser's research bridges the realms of numerical mathematics, scientific and high-performance computing, and life sciences, where he develops numerical methods and models to investigate the relationship between structure and function in biological systems. Today he'll be delving into the cutting-edge world of VR applications in bioinformatics and in neuroinformatics. Please welcome Dr. Queisser.
Yeah, thanks for having me. So I'll talk about NeuroVISor today, and until we get to the actual application, I'll go through the relevant biology, so you see what NeuroVISor is all about and what it's supposed to do. And as you can see, there are a bunch of contributors to the project. So this has been a project for the last 5 years or so, and has involved a number of undergraduate researchers, some graduate researchers and postdocs, and the project itself is headed by Dr. Seibold and myself. We're both at the math department. So everyone probably has at least heard about, if not used, VR headsets in the past, and they've been around predominantly in the gaming industry. But then in the research field, there are various research areas and departments that started using VR technology to augment typical lab experiments in medical research. It's used a lot in the context where the subject usually wears the headset and data is recorded from the headset and the interaction. And then in the end, researchers get together and evaluate data, analyze data, and draw research conclusions from that data. So the main tracks for VR are and were gaming plus this research component, which was data driven, so collection of data through the hardware, this novel hardware. And we had a somewhat different idea in mind when we were thinking about VR, and that was, is there a way to use VR technology not to record data and then analyze it later on as a researcher, but can we as researchers actually wear VR and use it directly in a research context so that we can study the research problem in and of itself within a VR virtual environment, rather than using it just as a tool, just in quotations, as a tool to collect data, which then later on gets analyzed. So this, if I can, oh, there we go. This brought us to the question of whether we can change a traditional computational workflow. So I I work in interdisciplinary teams most of my time. And what makes the process a lengthy process is that we get together as researchers from different areas. We understand each other's languages and try to consolidate it. From my end, we try to consolidate it in a mathematical language and framework. And then we have a problem that has to be solved computationally. So then we start designing a simulation, as you see in the box on the top left. So simulation design is the derivation of mathematical models, let's say, that describe reality to a certain extent. We take those models and we have to, typically they're complicated enough that we can't find closed analytical solutions, but need computers to solve the problem. So that means we have to then implement code, run a computation that can be on a local machine, but can be on an HPC cluster somewhere. So we send off a job, we wait till it's finished computing, we then bring back all the results, all the data from that computation, and then locally start analyzing the data, be it visualizing it or looking at time traces, etc. And then we have that picture and we come back as a team and start discussing the results. And then of course we have ideas, hey, we need to change certain parameters or we have to change the model. So we go back and modify the simulation, send off the job. So you can imagine this is and probably many of you already experienced, this loop is fairly long. It's a lengthy process to iterate, and then that iteration has to converge into something useful. And this is where VR technology hopefully can short circuit that to a certain extent. So the real-time interactive high-performance computing driven by VR technology, that concept has in mind that if we are able to implement a mathematical or computational model of what we're trying to describe in such a way that we can, in real time, visualize the results of a simulation, then we have a much sort of shorter loop to go through because it's intuitive, it's three-dimensional, and again, most of the typical applications are visualization of something static. So here we have additionally something that propagates in time. So three space dimensions, a time dimension, and this whole thing about gaining intuition and then modifying the simulation and running new computations, that should also happen in real time. So we should be able to interact with the simulation as it is running. We don't want to send a job off and wait for it to be finished and come back. So now we have the task of, can we solve our computational problem in real time? So that's a whole new high performance computing question. Why? Because if we're in VR, we want to update a solution roughly 100 times per second because we need that frame rate to not become seasick using VR. So where we could wait hours or days to compute something on an HPC infrastructure, now we have the demand, hey, you need to produce an update 100 times per second. And we want to make this interactive. And NeuroVISOR is a prototype of that idea. So we set out to ask, is this feasible in the first place? And we needed a problem that is not geometrically trivial. So anything that's geometrically trivial is maybe not very attractive in VR. And we need something that is complicated enough from a modeling perspective that we can see how far we can push the system to really stay within this real-time 100 Hertz scenario. So it can't be an easy mathematical problem and it can't be easy geometrically. And yeah, that brought us to the neuro part of this tool. So we looked at neurons since they have a complicated, beautiful architecture, and there's a lot of interest experimentally as well as computationally in that area. So the field of computational neuroscience is huge. which means there are many developed computational models which are non-trivial that describe signal processing in neurons that we can use. There are mathematical tools from our area that we could combine. And there is a very nice morphology of neurons in the lab experimental setting, a nice infrastructure that leads itself to this type of prototype. So I don't know, does anyone work with neurons in this group? Formally, but yes. Okay, so as you probably know, our brain consists of many, many brain cells. They're interconnected via so-called synaptic contacts. And what our brain consistently does is take sensory input and translates it into electrical signals and then downstream into biochemical responses. So neurons have a cell body, the soma, from which two tree-like structures branch off. One is called the dendritic tree and the other one is the axonal tree. They are historically separated into those two categories. Now that boundary between dendrites and axons is somewhat softening. But in any case, you can think of this as a tree growing in one direction, the roots growing in the other direction. The dendrites classically are thought of the part of the cell that receives information from the synaptic inputs, then integrates all that information and passes it on to the axon, which then transmits signals back to other connected neurons. And the signals we're talking about there are electrical signals. Now, these electrical signals then trigger biochemical responses that involve intracellular organization. So the endoplasmic reticulum that's filled with calcium gets triggered by electrical signals in the cell. That produces, for instance, a calcium response in the cell that then regulates gene transcription at the cell nucleus where the DNA is stored. So there's that interaction and that coupling between biochemical and electrical signals, which is what our brain, like I said, is doing all the time, trying to make sense of the input we're receiving. Now, on the other hand, that what I described verbally can be translated into model equations that hopefully describe the correct electrical signal processing or the correct biochemical responses, et cetera. And if we can do that, we can then go and compare to experiments that are at the cellular level. So what you see on the bottom right, this image is an electron microscopy image of a neuron. So you see the soma here. This looks like the soma of another cell and all those dendritic and axonal branches growing out of the soma. This thing here that you see is actually an electrode that is inserted into brain tissue targeting the soma, the cell body of the neuron. And that electrode can be either an electrode that controls the voltage at that particular point, or it actually injects an electrical current at that point. So there are current clamps and voltage clamps. This technique was invented, I believe, in the 80s and won the Nobel Prize in the 90s. And I fortunately had the opportunity of working with one of the Nobel laureates who invented patch clamp techniques at the cellular level. So this technique of being able to manipulate individual neurons and then recording the electrical response at that neuron in different locations really gave rise to quantitative understanding of what individual neurons are doing in the brain. So in short, the experimental lab setup for these types of experiments are take brain tissue, isolate individual neurons under the microscope, insert electrodes, and manipulate the neuron from the outside through electrodes, and perform recordings to see how they respond under different stimuli, which would then give rise to an understanding about individual neurons process information. And that's what we try to replicate in NeuroVISOR. So NeuroVISOR is intended to be the in silico virtual reality lab environment for these types of experiments. Okay, so I'll just run through the concept of deriving these computational models for what I described just now. So you have an understanding of what NeuroVISOR is actually computing in the background. So you start with Ohm's law, and a cell, due to its membrane properties, functions as a capacitor. And what we're trying to track is the voltage, the membrane voltage over time. So capacitance times the voltage is Q, the charge distribution. And starting with that equation, we can then ask, well, what's the temporal evolution of the membrane potential, that voltage? So dV over dt, assuming capacitance is constant, times capacitance equals the change in charge distribution over time. So how does that charge distribution change over time? It changes with ions moving from the extracellular space to the intracellular space, or vice versa. Or that would be the membrane current here, I sub n, or I have an electrode. So we're looking at either membrane currents or electrode currents that can change the state of the neuron. So now with this equation, we eventually have a differential equation for this quantity V, the membrane potential. At the moment, that's just in one location, equals the membrane current, negative membrane current plus electrode current. Okay, but how do we model the membrane current? So this is now a biological membrane that allows ions to move from one side of the membrane to the other side. So channels, pumps, all those biological components are embedded in the membrane that drive the membrane current, and the membrane current then is the sum of, well, this quantity GI times V minus E sub I. GI is the membrane conductance for an ion species I, so I can consider sodium ions, potassium ions, those are the traditional ones. Chloride, calcium, all the ion types present in the system can be described by one of these terms. And the motivation for this is that we think of this biological membrane functioning as a capacitor. So it can just store charges on either side of the membrane. And that accounts for the capacitive flux in the equation. So that's the left-hand side of the equation. But if that's all, then that would mean nothing can change over time, right? That would be boring. And in fact, the biological channels, pumps, receptors, etc., can be thought of as resistors in the membrane combined with a battery. So the membrane conductance for a specific ion species can be described by a resistor for, let's say, sodium, potassium, or chloride. And the batteries are this voltage, this reversal potential. So each ion has its own desired potential. So I don't know if you've used the Nernst equation in the past, but the Nernst equation describes for a single ion system the voltage at which it would like to come to rest. So sodium ions will have their own reversal potential, E sub sodium. But then potassium will have a different reversal potential based on its properties. And basically that has to do with the concentration gradients between the intra and extracellular space. And since there are different gradients for different ion species, their resting potential is going to be different. So now you have these competing resting potentials. That's why the term V minus E is called the driving force. So one ion is trying to drive the voltage towards its resting potential. The other ion species is trying to drive it to the other. The combination of which then governs the membrane current. So taking all of that together, let's see, can I go? back, so what you, oh, I think I have it on that. On this slide, yeah. So what we now have to understand is how does that conductance value, G, change over time? Because it's not constant. Channels open and close and allow ions to pass through these channels selectively at different time points. So this conductance varies over time. And this is where Hodgkin-Huxley in the 50s developed their Hodgkin-Huxley model, which again is a Nobel Prize winning piece of work. They figured out that sodium and potassium channels are the channels that gate sodium and potassium and are the main contributors to the formation of these so-called action potentials, the electrical signal that travels through cells. So they observed this conductance, this membrane conductance for sodium and potassium is not constant and it changes over time. And the way to think of it is, I know some people do molecular dynamic simulations here, so that would be the idea of really understanding a whole protein structure, a whole ion channel, let's say, and understand the detailed force fields that allow ions to move through these channels. Those simulations, as you probably know, require high performance computing and are on the scale of femtoseconds. Here we want something that's on the scale of seconds, minutes, hours. So Hodgkin-Huxley derived empirical models, so not driven by fundamental physics, but more of an intuitive understanding of what a channel does and then matching parameters to experimental data, and they agree surprisingly well. So the way a channel is thought of in this context is it's basically a tube, and that tube can be closed by a swinging door or a ball that can be shoved into the gate and therefore closes or inactivates. Illustrated here in this image. And Hodgkin-Huxley found that sodium channels are driven by two of these mechanisms, a swinging door and a ball that can activate, inactivate, open and close the channel. In order to simulate or model these two units, they introduced quantity M and H, which are these open gates. So this particle, if you think of it as a particle, can either activate or inactivate or open or close the channel. And there can be a number of individual units, M and H in the system. That again is something that has to be tuned to the experimental data. And those quantities are described by differential equations for M, for H, and the potassium channel is a little simpler. That only needs one gating particle. But all three equations are differential equations, and if we have the correct parameters, the time constants, the steady state values, etc., then we can solve those differential equations computationally. And those together now describe when a channel, a sodium channel or a potassium channel are open. So this is now the final equation. Remember, we have our capacitive current here. We have a maximum conductance. If all channels are open, this is your conductance for sodium, your driving force, and now we have these quantities m cubed times h. Those are the solutions to these differential equations. The exponents come from experimental data and tuning to experimental data. And the same for potassium. Here we just have one unit to the power of 4 times the maximum conductance. Everything else is dumped into the so-called leakage term. So any ion we're not explicitly considering gets thrown into that term. So now we have a differential equation in the voltage, as you can see. And to solve this differential equation, we have to solve 3 others. So if this neuron is just 1 point, we have 4 differential equations to solve. And if we do so, we can show what the voltage trace over time is given some external input. And that produces action potentials. So our neurons don't reside at 0 voltage at rest. That's due to the different ion gradients. Remember, the Nernst potential for different ion species. So typically a neuron will reach an equilibrium state at around negative 70 millivolts. And if I start prodding this cell by, for instance, injecting a current, I can move the potential away from the resting state. And once I reach a given threshold, my sodium channels start opening. Now those quantities M and H, because they're dynamical quantities, they change over time, and now all of a sudden that's going to produce a sodium current and change, depolarize the cell. But at the same time, the potassium channels start opening up. They're slower, and since their resting potential is way down here, they're going to start pushing the membrane potential back down in the other direction while the sodium channels are inactivated, because they have this inactivation component. And then there's a slight undershoot, this hyperpolarization, and then it comes back to rest. So this is a very typical waveform of the membrane potential at a single point if we prod it. And that you can think of as a wave propagating through the cell, starting at the axon typically. So this is sort of the unit of information that can be passed on. And all of that can be described by the equation above. Okay, but now again, we're going back, we wanted something complicated in geometry. So we don't want a single point neuron, we want something that, that resembles reality. So we can't just solve this problem I derived in a single point, but we have to find a solution on many points. So if we take a neuron reconstruction with a complicated shape, what we'll do is we'll subdivide the neuron into compartments. So we just select a number of points on the geometry. We put cylinders of a given length around that point. And now we have a local problem that is cylindrical with one unknown, and that's where we solve those 4 differential equations I've derived. And now those cylinders crosstalk, right? If the potential is high here and low here, then we'll have electrons flowing in that direction. So due to the potential gradient, we get a coupling term. And this is right, where is it? Here, yeah. So now we're looking at the difference between the voltages that of connected compartments plus the membrane current that we had earlier. So now we get a system of differential equations. For each of the cylinders that describe part of the neuron, we solve 4 differential equations. If we have 1,000 points describing the geometry of the cell, we have 4,000 differential equations to solve. So the level of detail in geometries will automatically increase sort of the set of differential equations we need to solve. And remember, we have to solve them 100 times per second. Okay. The next caveat is, how do we actually get this into VR? So now we have the equations, but where do we get the geometry? Well, there's one huge database, neuromorpho.org, that hosts about 250,000 neuron reconstructions from labs all over the world. Different species, human, mouse, rat, monkey, all different species, different parts of the brain. So it's a huge repository for neuron morphologies. But the only information that's stored on this database is a 1D graph representation of the neurons that have been reconstructed. So we just get points, 3D coordinates for each point on the neuron that was registered, and we get the connectivity pattern. So we know the edges between nodes or vertices, and in each of the vertices, we get the diameter of the dendrite. So the diameter information, the coordinates of the points that define the geometry and the edges. But for VR, we need a surface representation. So we need a surface mesh, a 3-dimensional object that we can grab and interact with. So from this data, we need to get to a 3D surface mesh. So we developed a tool that takes that original morphology tree, and as you can see in the top left, that would be that morphology tree with the diameter, using the diameter in each point and building little cylinders around it. So you see, that typically is a huge mess. What we want is a closed and watertight surface. And for that, we have to go through a bunch of steps. So there's a preconditioning step, a geometric modeling step, and actually the really tricky part are the branching points. You can imagine if individual branches from dendrites come together, these are all just cylindrical descriptions. Now we have to build something watertight there. So that's actually more involved than one perhaps would think at first. So we have the database with the 1D graph information. Now we can translate that to 3D surface meshes. So now we have together the geometry and the model. But remember, the model itself is formulated for that 1D geometry. So in fact, what we're doing in NeuroVISOR is we're using both. We have a 3D representation of the geometry in NeuroVISOR. And hidden in the background is the corresponding 1D graph on which we solve this ODE problem, which could consist of 4,000 or 10,000 or 100,000 ODEs that we have to solve. And then map the solution onto the surface mesh so that we can actually see what's happening and we can then interact. So interaction too is an interaction with the three-dimensional geometry that then has to be mapped to the 1D geometry where the problem is solved and mapped back as a visual representation. So these screenshots here, and for everyone who later on is interested, but in general is interested, we have a lab with the hardware and the tool running. So if you want to try it out, let me know because it's way more fun than looking at slides for this. But we reconstructed our lab, and on one side of the wall, we have a cell selector. So you have a visualization, a 1D visualization of neurons that we can offer for simulations. So that's the top left. And once selected, we have a control board where we, for instance, can inject currents directly. So that interaction mode direct is just a ray cast onto the geometry, and wherever you ray cast, you're injecting current and watching the response. Clamp mode is, like I said, the experimental, virtual experiment of placing voltage clamps or current clamps at different locations. And then the plotting mode allows us to open up windows at specific locations to actually see the trace of the voltage over time, so that we have not only the color-coded feedback, but actually the data feedback of what's happening at that particular location. So here you see our clamps are these discs that are placed at whatever location you choose, and they trigger then a response in the neuron. And we also have synapses, so we can not only simulate on individual neurons, but we can grab multiple neurons and start connecting them synaptically, so we can simulate networks. At the moment, admittedly, we're looking at very small networks on the order of 10 neurons, which is not comparable to our brain, hopefully. So here you see one of our undergraduate researchers using the tool. So that's our lab. We have a TV monitor, so if someone's using the VR headset, other people can stand by and observe what's going on through the monitor. And here you see a typical scenario of a neuron that has one or multiple clamps placed. The color coding is now the membrane potential, the voltage at different levels. So you see here the scale at the moment goes from 0 to 50 millivolts. So red would be sort of the most depolarized state before the cell hyperpolarizes again. And we see those voltage windows that show us the potential traces at individual locations. Okay, here we have, I think this is a video. Yeah, so you see with the controllers, you have those controller hands, you can raycast onto first the selection gallery. So the user here is selecting this cell. Once selected, it appears in the room, and along with its control board, You can grab it, you can move it around. There's a ruler, as you can see, so you can rescale the size of the cell if you want to. And once placed in the lab environment, now you can raycast a current, in this case to the soma, and now you see the action potentials propagating through the geometry visualized by the change in color here. And this is happening in real time. So again, this is 100 Hertz simulation. The solution is plotted 100 times per second. And therefore, the computational code in the background has to be optimized to really carry that task. So here the user placed a voltage clamp, so you can place it and step back. It's held at a specific voltage. And now you see this very typical rhythmical action potential propagation through the individual dendrites or axons. And depending on the input, of course, that changes, and that's exactly what lab scientists do at the cellular level. So now we can go in and start modifying parameters in real time, modifying experimental conditions in real time, short-circuiting that typically very long loop to the cell. Yeah, now one of the visualization windows is open. What's happening at that particular location. Yeah, so I'll close with this last example. So this is a so-called Purkinje cell. You see the branching structure is such that it gives rise to very intricate waves within the cell. So you see different oscillations occurring in different parts of the neuron. So you start seeing that the problem, due to the complexity of the geometry and the underlying equations with their parameters, it starts becoming unintuitive if you just write it down on paper and try to figure out the dynamics. So you do need computation and simulation for these types of experiments. And even better, if you can do it in real time, you can modify and really see what happens when you do certain things. Yeah, so that's NeuroVISOR at the moment. I mean, there are a few more features that I'm not highlighting today. Feel free to stop by. Again, it's a prototype, and a prototype that could also be moved towards very different applications. So us as mathematicians, we're thinking about, is this even something that could help us understand mathematical algorithms? So we implement different solvers for PDEs or ODEs. And we learn all the theory. We write down and prove that this is going to converge and work under certain conditions. Once you bring it to these types of applications, well, a lot of the things we assume when proving certain things don't hold anymore. So we're not quite sure how the methods are going to behave. If we can test them out, this is very intuitive. If we could compare two solvers and say, okay, this is how this one is behaving versus that one, and we get some feedback, it could even give us mathematicians an idea or an intuitive understanding very quickly of different mathematical methods. But then again, of course, biology has various directions one could go there. So this, again, it's a prototype. It works. A lot of undergraduates and graduates were involved, and like I said, if you want to try it out, or over there in Walkman Hall, just let me know. Okay. Yeah.
I'm curious what the resolution that you can simulate is, like how many ordinary or partial differential equations can you solve per second before it becomes too much for your simulator?
I don't have an exact number, but we have tested the system on the order of 10,000 degrees of freedom times 4 for the ODEs. So somewhere on the order of 40,000 ODEs that we can solve at that speed. The GPU and CPU simultaneously. So the GPU is reserved for all the visualization and interaction. That's multi-threaded. And then the computation itself, we run separately on the CPU. So there is a level of parallelization Also rudimentary still, so we can push it to that order of degrees of freedom. But then if we look at larger and larger networks, for instance, that's where we're currently looking into, well, now you need a higher level of parallelization along with the numerical methods we implemented.
By larger and larger networks, do you mean larger neurons, or do you mean the possibility of seeing how impulse might travel from one neuron to another across the synapse, for example?
Right, yeah, so larger systems I'm thinking of typically just larger networks. So going from 10 neurons that are interconnected
Right.
to 100, 1,000. But then the other direction could be I need a very fine resolution of a single cell where I can't represent it as 100 points, but I need 10,000 points for whatever reason. Either the geometry is so complex or I need that resolution to be accurate enough. So both directions, it's just a scaling of geometric points that we have to solve this system of differential equations on.
So to kind of piggybacking on both of their questions, what level of how far should we go? Maybe this is more for a neuroscientist to answer, but you obviously you know what you're doing here. How far do we go in scale? Is it the level of like channelomix and ion species before we kind of start abstracting away unnecessary details for, like, say, medical XR, medical VR purposes?
Yeah, so the models I described here for the electrical component are surprisingly accurate for many, many cases. So the level of detail that you typically add to that is the same idea as what I proposed. So it could be additional channel types. So instead of just sodium and potassium, now we have 7 different calcium channels and chloride channels and whatnot. But they fit this framework. So we're not changing the modeling concept. And you can get far with that. Now, the next question towards sort of medical or neuroscientific application, what are we trying to read out, becomes a question of, is it just the electrical signaling or is it also the biochemical signaling? This is actually a direction I would like to go in terms of calcium dynamics, because calcium dynamics are involved in many, many processes. A lot of like the modern topics we're researching, Alzheimer's disease, Parkinson's disease, all these things, learning, plasticity, all has to do with calcium. So that's where I would like to go in that direction because it has big medical implications. But now you're talking about a level of detail that goes beyond what we're seeing here, because the intracellular organization plays a critical role. So now we have to resolve the whole cell in 3D. We need the intracellular organelles like the endoplasmic reticulum, the mitochondria, the cell nucleus. Those have to be resolved to really understand the dynamics. Now that's a whole different model, still far from like single channel or protein level dynamics, way, way beyond that on a time scale. But that's, I think, the next sort of area one would have to push towards in expanding application for this type of a tool.
Is that route your current primary interest, or are you more interested in expanding past 10 to 100 or 1,000 neurons?
It's both. So at the moment, it's pushing complexity and just trying to break our system as we have it. Just see what can we do to break it, and then see if we can fix it. And like I said, going towards the biochemical application side, that opens up a whole different architecture that we have to think about. And it's also a different time scale. So here we're talking about millisecond dynamics. Now if we're talking calcium, we're talking about minutes, maybe even hours, because it's all much slower in turnover. So, and we've been putting together ideas of, well, how would you bridge timescales? We have dynamics happening on the millisecond time scale. We have processes that are relevant on the minute time scale. Now, real-time computing, what are we visualizing?
Can we push certain things in the background, bring other things to the foreground? That's still quite hypothetical. So our team currently, with all the undergraduate students involved, they're more focused on how far can we push it currently, and once we've broken it, how can we prove that we can get even further in network complexity?
So speaking of that, what is the limiting factor on this? What's the main barrier?
It's the computational cost for solving the differential equations. Yeah, that's the bottleneck. Yeah.
So with the quantum computing, that whole revolution, is that going to help this? Does it have potential use?
Yeah, I think that'll help, in theory, everything.
Right.
I mean, one much smaller step would be, for instance, to, instead of the CPU, put in an extra GPU and try to solve those problems on the GPU. GPUs are great for anything that's structured. That's why image processing, et cetera, that's always great. If you have a structured problem, GPUs are wonderful. If you have an unstructured problem, and when you deal with PDEs, you start getting unstructured problems. So the set of ODEs I've been talking about are actually a consequence of solving a PDE in space and time. So the moment things become more unstructured, getting the most out of a GPU becomes more and more difficult. So, but that would be, I think, before we jump to quantum computing, that might be an intermediate step. Yeah.
Is this compatible with the HTC Vive headset?
So our lab has developed everything for Oculus devices. However, the library has various models, and I believe a year or two ago, they tested it on an HTC. And I think it worked back then on that device. But there are some fundamental challenges, like covering all types of headsets. So since we're focused on the research of the tool, we've focused on Oculus devices because it's easier than having to support, or making sure it supports all other devices. But the Vive, I think, at some point at least worked, and we had last semester, in the spring semester, for one of the neuroscience courses, we ran for the first time a prototype project within the course where students use NeuroVISOR for their lab experimentation, and that was done at the VR studio in the library. So that was.
I wish I graduated from neuro later, so I got a chance to test that out. That'd be cool.
Well, it's like I said, it's over there if you want to try it out. And there is a desktop version that is fully operational under Windows. Mac, again, causes lots of problems. We got it. We have a build that works partially, so not the full feature set. But if you have a Windows machine and want to try it out in the desktop version, there is a desktop version. Again, definitely not as fun as the VR application.
Touching on that CFD bit we were talking about before your presentation, just like a light kind of thing. Have you ever considered, there's some research I was seeing about modeling mesh-based arterial networks. So for much larger than a single neuron, but there's a lot of them empirically that appear in close proximity to arteries, namely astrocytic cells.
Mm-hmm.
So I'm curious, is that a potential direction that intrigues you, or not really?
I think I find too many things intriguing, but yeah, I would.
Like feasibly?
Yeah, so I mean, the arterial system, in a sense, is very close to what we're looking at here. You have this branch structure. There are tools for geometry, surface reconstruction in that area of research that are sort of comparable to what we've been doing. And also the modeling approaches are similar in the sense that if people today try to simulate sort of the entire cardiovascular system you can imagine. That's fluid dynamics. That's a lot of computation if you wanted to resolve the entire cardiovascular system in its entirety, 3D fluid flow. So what people have been doing there too is develop reduced models, so parts of it, or even vast parts of it, can be modeled as these one-dimensional reductionist components, like the Hodgkin-Huxley equation on a graph, and then only resolve where necessary the three-dimensional details of the system. So there is this hybrid dimensional idea that is being used in that area of research, which also makes a reappearance in neuroscience because of the similarity of the geometry. Very different models. The one is electrical biochemical, the other one is fluid flow model. But yeah, I think there too, VR would really open up a lot of possibilities of an interactive environment. However, they're going towards medical. So I talked to a colleague a while ago, and she was telling me they had for medical planning actually developed VR-based tools for surgeons to perform pre-planning in their surgeries, and they had this all developed, the mathematics, the VR, everything. It was beautiful, and then they brought it to medical clinicians, and they refused to use it because they look dumb in a headset. So there are like these very unpredictable components, like no, people just don't want to use it because they don't feel comfortable wearing a VR headset. Things like that pop up. So not that that should hinder anyone to develop these types of tools, but it's somewhat surprising sometimes to see what barriers exist that you don't expect at all.
They gotta get the HTC goggles.
Right, I mean, technology will change and it's
Yeah.
going to become more and more lightweight, but it's hugely interdisciplinary, and there are a lot of components also from sort of the more soft science areas to consider. Like originally we had our lab designed, we're on the 10th floor with the lab, so one of our students took a photo from out the window onto campus over here, and then used that photo as the backdrop of the window in the VR lab. And then we had people from psychology test the tool and said, well, some people might be afraid of heights and the 10th floor is too high. So we replaced it with a photo from a floor on the 4th floor, I think, or 3rd. But those are things, at least I, as a mathematician, then don't necessarily think about when designing these types of tools. So it becomes even more interdisciplinary than one might think.
Do you use, I think I stepped out when you were speaking, so I apologize if I missed this. So do you use at all the leaky neuron model, or any other types of models besides HH?
Yeah, so I guess the leaky integrated fire models are a simplified version of Hodgkin-Huxley because they don't account for the detailed sodium and potassium dynamics, but are just a capacitor-based model. So this capacitive current, you get a depolarization up to that threshold that we saw when an action potential is formed. But anything past that threshold, which is driven by sodium and potassium channels, is no longer covered by the leaky integrated fire model. So all you can model is up to that threshold, the depolarization. And if you know the threshold, then you can say, well, that's where an action potential occurred. So I'm going to mark an action potential and then reset my system to the resting potential. So a leaky integrated fire model can predict the occurrence of action potentials, but not the waveform of it. For many applications, that's enough. So you wouldn't have to go through all the Hodgkin-Huxley based computations, which would make it simpler from a computational perspective. And yeah, going towards larger and larger networks, maybe that's a compromise to make if you want to go to 100,000 neurons. Maybe not all of them are resolved at that level of detail that I showed today.
Well, thank you so much for coming and giving it
Yeah,
today.
thanks for having me.
To quickly just
questions from the TV, and if anybody else has any questions, feel free to go on up.
Can I ask you one more thing?
Yeah, sure.
So the arterial paper I was referring to was an archive preprint by this guy named Pim de Haan. He works in Amsterdam with a lot of the, you might be familiar with like geometric deep learning or training and testing on 3D objects. So the paper was called Mesh Convolutional Neural Networks for Wall Shear Stress Estimation. It's like a phenomenon in embolism in medicine. And I'm just curious, like, is there any type of AI you might leverage to, say, predict instead of getting a novel neuromorpho file from a 1D array, instead you could take the three-dimensional thing that you have it manipulatable in VISOR, and then take it out, run a data set or whatever, one shot, whatever, such that you can predict how it would evolve without needing chromatin dynamics or putting more ion channels in there and studying the axon.
Some of the collaborators all around.
Yeah, absolutely. I mean, that's,
That little thing is constantly watching.
I am Big Brother.